Okay, if there's are no questions, I think Luke, you can move to our next presentation. So Luke is gonna give you also overview of our sample environments, and then after Luke's uh, presentation, we can have a break.
All right, thank you, Wellington. All right, so the next part, I promised I was going to talk about sample environment, so here we go. I mentioned this sample area in the tour. It's all within this black aluminum frame right off outside the tank. It is very adaptable. It's about 4 feet wide by a couple feet deep. And we can break down on the left here. Let me switch to a different pointer. On the left here, we've taken out most of the equipment inside there, so you can see that it can actually break down. And we have this beam extender tube with an aperture holder on the end, so we can change the aperture size at that point. We can change the beam extender tube to different lengths, depending on how big the equipment is that we need. But a very typical setup for us is we have these two motors installed, and the one on the left here, which is closer to the beam source, this has this kind of zigzag piece. That's our aperture changer. It has 4 different size apertures. This one goes from 10 to 16 millimeters in 10, 12, 14, 16 are the aperture sizes. So we can shift that through the beam and optimize the amount of beam on your sample. And then the front motor here will change. We'll have a sample changer of sorts, or hold the actual sample equipment that we need to use, and that also moves through the beam. So if you have multiple samples, we can shift it through the beam. So we'll get to that in a second, but first I want to talk about the sample cells that we use. So I'll start here at the top right. We have our typical banjo cell or Helma quartz cell. These are purchased from the Helma Corporation, and we have a vast supply of these, but users are also able to bring their own if they want to. Our typical sizes that we have are 1, 2, 5, and 10 millimeter path lengths. So that is the distance from the front face of quartz to the back face. And just for a volume estimate, the 1 millimeter path length is about 300 microliters volume. So that scales up with the rest of them. Typically, you'll be using the 1 or 2 millimeter path lengths for samples on BioSANS. The next common sample cell that we have is the titanium cell here in the top middle. This is something that is considered demountable. It has a series of O-rings, windows, and a spacer in the middle that you can also do 1, 2 millimeter spaces. And you can assemble these with a solid sample inside, or maybe a very viscous solution, and assemble them in place, something that won't go into a banjo cell. And then the screw on top is a solution filling port for you to add water or D2O or whatever solution you might need after that other sample is loaded. This is used for a few different of our sample environments, and it's something we've tried to ship out to users before, but it does have the center core sealing nut that compresses and needs to be torqued to seal properly. So we don't prefer to ship these out, which is why we have now developed this faceplate cell, which is the next one here to the left. This one is also titanium, and we have 1 and 2 millimeter spacers for these. The difference here is it has these titanium plates that go on top instead of a center core, and these can be sealed using these 4 Allen wrench screws. So a little easier to use in home institutions. And also because the faceplate will go on without twisting. If you have something like a fibrils or something that needs to be aligned when it goes in, then it will not twist the sample when it gets sealed as well. Next, moving down below the titanium cells, this is our pressure cell, or EAP for extended angle pressure. This is another thing that you need to assemble. If you're using the pressure system, we will require you to be on site. We have a setup in that general chemistry lab where you can put these together. They have large quartz windows and double O-rings on each side to contain the pressure. And then a sealing ring around the outside that has this cutout for the angle for the scattered neutrons to leave the sample and actually reach the detector. Those are pretty much the main stuff we've been using at BioSANS for sample containment. But there are a few other options. On the bottom here, we have different Helma quartz options. This is that same banjo cell. And then there are rectangular cell options that if you need more height, or if you are bringing your own, we can accommodate some of these other cells. There's multiple different sizes. If you're going to use anything but the banjo cell and titanium cells, we prefer you talk to us first, just so we know what's coming and maybe what we need to accommodate if we can actually fit it into our systems. The last two things with sample cells is in the bottom right corner here, we have a new sample cell cleaner. So typically in the past, you've always had to clean the cells as you leave once your experiment is over, and you do this one cell at a time. This multiple cell cleaner can actually, these 4 orange O-rings down here, you can put 4 banjo cells on there and flush them with ethanol and DI water to clean out the cells and do them automatically 4 cells at a time. That'll hopefully speed things up. And then we also have these little 3D boxes that we've printed that will hold up to 12 banjo cells. And as you can see in the banjo cell, we have these little caps that go inside, and then the little hats that go on top of those. When you put those all in here, then this plate goes over the top, holds everything in place, and pushes down on those caps to make sure everything stays sealed. And then once the lid goes on, it keeps all of that in place and screws down with some thumb bolts. That keeps everything nice and tight and sealed for shipping. So this allows us to ship some banjo cells to users for filling and remote experiments. Again, if you're going to do a remote experiment, please talk to us ahead of time and we can work out the details of that. So going back to the sample environment, I've mentioned our sample area and the motors. We have several shifters that will mount to those motors, and a lot of our equipment has a general cooling and heating capabilities. So we have this water chiller up in the top right here, and that can go from 4 Celsius up to 80 Celsius. And it has large hoses that go into a base plate for several of these shifters, as well as some of our other equipment. So it can control then these shifters down to roughly 10 Celsius or up to 80 Celsius, depending on how well they're controlled and how much mass is in them. The shifters that we have that mount directly onto those motors and this chiller control are the banjo cell shifter, which can hold 15 samples, our titanium cell, which has 9 samples, the rectangular Helma cell that can do 18 samples, and then we also have a stretcher that can pull a solid sample. When you're cooling with these, sometimes there's condensation. So this white box at the top here is actually allows us to flow dry air over the banjo cells and keeps the condensation off of those banjo cells while we're cooling. Next is the robot sample changer, which you saw in the tour. What this is sort of our workhorse sample environment now. It is a 6-axis mecha robot and has this gripper on the end. There are spaces for 48 banjo cells to be held in these storage compartments around the robot, and they sit in these red baskets where the robot can grab those baskets and pick them up and move them into up by the sample position here, which is a closer image on the bottom left here. And that sample position has a Peltier attached to it, so that can also go from 4 Celsius up to 80 Celsius for a single sample at a time. And the samples can be changed in about 45 seconds. So that is pulling the sample out of the sample position into storage and then taking a new one from storage into the sample position. This really cuts down on some of the effort to change samples. If you can compare even just doing a rack of samples, you're going to spend several minutes changing a rack out and putting a new one in and turning the beam on and off. So this will help you if you have a lot of samples ready to go. Or if, say, you have a large contrast series, you can prepare all the samples ahead of time and put them in here. The storage area does get chilled by that water chiller, so it can go down to about 15 C to keep your samples cool while you're waiting for them to be run. And then you would individually heat the samples when they're in the sample position. As Kevin mentioned in his intro, he's been working on the SEC SANS, which is size exclusion chromatography. And this is an image of it here. It's a large setup where we have these HPLC pumps that go into this chromatography system, and that allows you to determine the size of the to do some size estimates of what you have in the solution. And then we have it feeds through capillaries into the stop flow cells. There are 4 of them, which you can see a better image of that here. Again, this mounts on that shifter. And so you can have 4 samples in there. If you're using this for something like a contrast variation series, you can easily get about 2 days of beam time out of just 4 samples, measuring different contrasts. And what you're looking at here with these cells is you've got this, it's actually a peak cell with aluminum faces on it to get a good seal. And we can flush in solution through the bottom and then out through the top. So you can put your sample in here and change the, you can change the solution ratio. You can change the contrast of the solution, the amount of D2O or D2O and water in the system. And that can allow you to do these contrast variation or different solutions. And this is going to help more accurate SANS measurements of stuff like dynamic protein complexes, which you can see up here in this graph where you have, this green is the measure in a size exclusion chromatography, whereas the blue is just a SANS measurement. So you get a little bit of this size limitation that you can exclude by changing, varying the solution. And also we're working towards accurately determining the SEC peak with fraction in the beam. And by doing that, you can definitely optimize your measurements. If you want to know more about this system, I suggest talking to Kevin Weiss. Doing one over at Brace or contacting him after the workshop here. So the next sample environment that we have is our precise temperature control. This is our Neutron IQ from the Quantum Northwest company. This has 8 sample positions that are all individually controlled through Peltiers. So each position can go from -10°C up to 80°C, and they are controlled within plus or minus 0.1 degrees Celsius. So this is the very fine control where those shifters are probably more half a degree to a degree between the samples. This is each sample at 1 degree. Or within 0.1 degree, and you can do 1 sample at room temperature while 1 is cooling or 1 is heating. And that way, while you're measuring 1 sample through a series, the next one can stay cold while you're in a safe state. This takes the banjo cells 1 or 2 millimeters, and you take that banjo cell and put it into one of these large blocks. The block adds a little bit more mass, which allows that fine temperature control. And it then slides into one of these positions and bolts into place, which gives you a good thermal contact with the Peltier stage. So 8 samples, you can do different temperatures to buy a lot of time and give you enough time to prepare the next samples that you'll be needing. A science case for this is one of our users using antioxidant vitamin E as a membrane RAF modulator. And what they were doing here is looking at the different tocopherols at different temperatures. And you can see in these plots that at the very coolest temperature, you're getting a larger intensity. And as they warm them up, you're getting lower and lower intensities. The difference between the light blue and the darker blue here is just 3 degrees. So being able to control within 0.1 degrees gives you enough information there that you're seeing here in some of the difference, significant difference between 2 of those temperatures. And then even going to the higher temperatures, you can see a larger difference. And so the temperature requirement here was required for something like this, where you're actually looking at these membrane rafts and trying to find the different intensities of those domains. Going back to the extended angle pressure reaction cell, I mentioned this as our one of our sample cells. So in this case, you load the sample in here with these large quartz windows, and this can then loads into a sample changer where we can do 4 different individually controlled um, pressure cells, and they go up to 1 kilobar and up to 200 degrees Celsius. This is also a very large setup that has a cart with all the individual temperature controls here, so you can do each one at a different pressure and temperature while you're and vary through them, so you can actually go to one pressure while everything else is waiting. A science case in this one is these in situ thermochemical pre-treatment of biomass, where as you can see the temperature increase, the pressure increase on these switchgrasses increase the intensity through the sample. And you can see that this asymmetric scattering comes out of this with the different temperatures as well. So this is something that we actually do control the temperature of the system as well as thereby increasing the pressure. And then here you can see some of fibrils, some of the biomass being in the sample cell itself. So this would be the neutron path going in, and then the neutron path coming out at this darker flange here. Another popular sample environment that we have is our tumbler. This is good for precipitates and colloidal systems. It can go up to 100 RPM, where you load the samples into a titanium cell, put it in this little cog sample holder, and then you can put up to 4 samples onto. It's a screw drive that runs underneath here, and then you put the cover in. This can be temperature controlled as well through the chiller. Because of the thermal contact, it's only 15 to 60 degrees Celsius. But in this case, there are some places where one of our users has found that there are these nanoplastics in soil, and as you're using some small plastic, small nanoparticles, they do stay dispersed in the water and don't necessarily need the tumbling. But as you get to larger nanoparticles in the 300 to 1000 nanometer range, they start to fall out and precipitate out. So as you tumble, and we would help find the optimum speed to tumble at, it keeps those large nanoparticles falling into the center and keeps them in the beam, so you can measure that sample, that particle size. And this was good for these convective transport, which led to the reduction of size of agglomerates in the system. So they actually did the study on both USANS and BioSANS to get a full range of data. The last equipment that I'm going to really talk about that the instrument owns is our humidity chamber. This does a relative humidity of 0 to 100%. It is becoming a more popular equipment with several requests on different beamlines to kind of go through measuring humidity on systems. We can control the flow and the temperature of that flow going into our cell here, this chamber. And also it takes a solid sample that has a rotational control. So when you open up this chamber, this is what it looks like here. And this has a sample mounted in the center, which can rotate the rotation stage is vertical, so you're rotating in the beam. And so we can control the temperature of the air flowing into the system and up to 100% humidity. Now, one of our users has developed a user supplied option for this, which you can see here, that does 10 different samples. So this takes out that rotational control, but you mount all 10 samples in line, and then you can control, and you can see here her samples mounted through the windows here with transverse and radial longitudinal alignment. So she was able to mount those, measure the humidity across all 10 samples, where you measure them all in line, change the humidity, measure them all again. So in this case, they were looking at forest products. So if you have these fibrils that come from trees or plant products, and you align them in the beam, and then measure them, you get the scattering a decreasing intensity with decreasing humidity. And from this, they determined that the spacing between the 5 rolls would actually be changing with that decreasing humidity. And we're able to convert this data into a spacing plot from 25 up to 96% humidity in this system. So a very useful system if you need to measure the moisture in your samples. Now, with user supplied equipment, we do accommodate users, and we'd like to work with your specific needs. As I mentioned, if you were going to use a cell that's specific, please contact us. If you have certain equipment you want to use, also please contact us. So some of the devices that we've used in the past with Vortex Fluid Dynamics, these are all images of these equipment off the beamline, but this has this large tube that rotates and creates a vortex solution. And then you can measure the particles in that solution in situ in the neutron beam. There's some actually interesting studies using these that if you're interested in some food science, they've done some measurements with eggs, and we've tested with some of that group on trying to get some biosans measurements on performed. Now, another one is an ultrasonicator that some users have developed and brought to do some measurements. What this does is it mounts to the top of our titanium cell, and this holder mounts onto that stage in the beam. And then this is an ultrasonicator, so it ultrasonicates the solution inside that titanium cell, and then this box is their Arduino control box. So this allows us, connects to a power supply in our system, that we are able to remotely turn on and off. So you can actually, they were ultrasonicating for 15 minutes and then turning it off and measuring and turning back on and off throughout the night. So it worked quite well, and we're planning to do some more of those measurements coming up soon. The last of the user supplied equipment I'm going to talk about is a flow cell. So the SEC system will kind of take place of some of this, but this is a quartz capillary or quartz rectangular cell that has ports on the side so that they can hook these capillaries up and flow solution through the cell, and then measure what's changing in the cell, and actually scan on a vertical stage this up and down to measure the different layers throughout that flow cell as they bring solution through. The last thing is our future development. So we are always developing, and some of the things that we have here are the titanium cell robot. So this is identical robot to our banjo cell, except we are trying to put the titanium cells in here. This is very close to complete. We are just getting some final production on our storage chamber for the titanium cells, and this will hold 56 samples at a time. So hopefully that'll help us use the titanium cells in a larger production. One other thing we've explored and some users have asked for is a rheometer. This is not something we've had on the beamline yet. This is a picture of it on the GP SANS beamline, but we have some designs to try and get it into the biosans if there are user requests to do so. Some of us are interested in developing an electrochemical cell where we can do in situ electrochemistry, apply a voltage to anode cathode inside a neutron beam in situ, and measure how the materials, how maybe the bulk solution is changing with that electrochemical application. And then there's a couple other things where we're developing with NCNR, which is NIST, a pressure system that can be uniform across different neutron facilities, and also we're exploring how to get to lower temperatures. So right now our limit is minus 15 C, but we've had some requests to go down to as low as minus 30. So we're exploring those opportunities. So with that, if there are any questions, I'll be glad to answer them.
Do we have any questions? Oh. Okay, I would like to ask everyone to turn their camera on. So we would like to take a picture for us to report this workshop, and then we can move for a break of 15 minutes. Is everyone there? You're missing a few
While
people.
people are turning on their camera, can I ask a quick question, Luke?
Yes.
So the robot loading the banjo cells, when I was there, I don't believe that was there. Is that newish, or is that kind of?
So um, that is finished. Can you hear me? My
Yes.
mic just switched. Okay. It was operational at the end of 2019, and we were able to use it quite a bit during shutdown and pandemic to bring in remote users. We did have a temperature control issue with it early this year. So I think that's when you came, right? Earlier this year. So it was out of operation for a little while, but we do have it back up and running now.
Is that the standard way in which we're moving cells around now, or are we still, turning off the beam and going in with our hands?
We can do either one. So it more depends on the sample cells that you need to use, the temperature control you need, how many samples maybe you have prepared. So if you're only doing 6 samples at a time, then sometimes the shifter is just easier because you can bring them in and load them, and the robot does take, because you have to load them in a basket and make sure you have all the positions correct. Sometimes it's a little more complicated, but usually it's fairly straightforward.
Thanks.
All right. I think you have everyone there. Great. Yep. Okay. Thank you, everyone. It's done.
Okay,
So we'll be back 11:25, and then we resume from there. Okay.